All right, so our next presenter, Russell Huie. Let me say that right, Russell, right? Okay, thank you. Another little slice of context, just referencing back to what the last number of presentations were. Russell's going to come and talk to you about the spinal cord injury open data commons, or SCI ODC. And the part of the reason why we wanted Russell to come representing the SCI ODC is because we've been after them for a while to help develop an open data commons for spinal cord stimulation. And I think after listening to the last number of sessions, you understand why, right? That there's work being done. There are different dogmas about how that work is being done, different approaches, different opinions, and the community, as you heard, I think is very, there's a lot. We'll talk about it later and tomorrow. There was a lot in this panel that you heard from the folks with SCI. But the whole idea is that how do we organize this information to lend itself to the translation and the potential translation or the real potential for these strategies. So with that bit of context, Russell, welcome. Please welcome Russell Huie.
Yeah. Can you hear me? Is this thing on? Oh, it is on. Thank you, Matthew, so much. This is just a great symposium. I have to say off the bat, I have not been here before, and I'm excited already to come back in the future. This is, I go to a lot of conferences with a lot of science and not a lot of advocacy. So this is really special and a really great group. So I'm Russell Huie. I'm at the University of California, San Francisco. Also hold an appointment out at the San Francisco VA Medical Center. And we're going to talk about data sharing and data reuse and data management. I'm going to make sure you've earned those free drink tickets. No, this hopefully won't be like too dry, but just a little background. So I came up, I was trained as a basic spinal cord injury researcher, preclinical sort of basic research that I had done for years. And I came out to San Francisco and have been working with Dr. Adam Ferguson, who some of you may know. He and I have been working together now for like over 20 years, which is gross. But it's no, he's wonderful, and he sort of turned me on to this idea. I came out just to do regular kind of bench work. And I realized very quickly that while that's very important, there's a lot we need to be doing with the data that we're actually collecting. So that's what I want to talk a bit about today. Let's see if I can. So essentially what we have in neurotrauma in general, it is, we can sort of think about this as a big data problem. And what do I mean by that? So some of you may have seen this figure before. This sort of shows the reality of what we're looking at, not just in spinal cord injury research, really all biomedical research. And what we're looking at here is essentially, we have a small number of data sets that are pretty large, that are really well organized, really well annotated. But as we watch this sort of slope go down across the number of data sets that are out there in the world, the size of those data sets get much smaller. The organization it becomes less and less. There comes a point where you actually reach the literature limit, which means beyond that point, this is data that is just sitting in file drawers, right? This is data that was potentially collected by researchers out there, but was not important to, be analyzed and put into your short, pithy, Nature or Science paper. There's a lot of data collected, and that data is sitting there not being used, and we're not getting anything out of that data. A colleague of ours, Malcolm McLeod, years ago was talking about this, referring to this as the long tail of dark data. And if you want to put numbers on it, I think he estimated in all of biomedical research, we're looking at about 85% of all the data that's collected is sitting in a file drawer somewhere, and that amounts to tens of billions of dollars a year. So this is important. And so this also gets to the fact that if all we're actually publishing is this really small, maybe 15% of the data we're collecting, we're sort of creating bias inherently by doing so. And of course, you know, then that leads to this idea of a reproducibility crisis. So that's like the bad news. The good news is, though, that people have been thinking about this and caring a lot about this for a while. And certainly funding agencies such as NIH and others are acutely aware of this and have made sure now to, this was, I guess, 10 years ago now, memo coming out of the NIH saying, hey, if you're going to get money from the NIH, you're going to do work with us, we need to make sure that the data you're collecting is being shared, which is a wonderful sort of sea change. Of course, it took a little while. And now here we are in 2023 with this NIH mandate that any of you out there who are researchers who are writing NIH grants probably in the past 8 months know that you are now adding this new data management and sharing plan, which is a great thing. So basically, we're making sure that the data that we're collecting, it lives on. There's sort of a legacy to the data. Okay. And that's where we come in. This is a little small, but for instance, if you're writing a data management plan for your NIH grant and you want to go decide, I'm doing spinal cord injury research and I want to know what's the best repository for my spinal cord injury research data, if you look that up, you will find us. So we have been developing over the past 10 years or so what's called the Open Data Commons for Spinal Cord Injury, odc-sei.org. This is what our homepage looks like. Anyone can go and sign up. It's very easy. This will be a short little pitch for it, but you go on there, you give us an email address. I will get a thing on my phone that says, this person wants to join, I approve you. And then you can come in and check it out. This is free to use. It's great for researchers. You can sort of manage your lab in there. But everyone can use this because one of the things that we are interested in doing is creating a system by which data sets can be published, just the same way that your journal articles are being published. The actual sort of underpinnings of those of that distilled information, the actual raw data should also be published as well. So you can go on to the ODC-SEI and click Explore Public Data. This is just because obviously we've been talking so much today about stimulation. I just want to use this as an example. One of our great postdocs, Anastasia Keller, had a Nature Communications paper a couple of years ago. She did some fantastic work at Louisville with pediatric spinal cord injury. So one of her papers, the data set from that paper has now been published, and you can go up there and peruse it. And of course, if it's human data, this is all de-identified data. Basically what happens is it looks very much like you're actually looking at a journal article. It has its own title. The title is very descriptive. It usually is kind of a long title because it wants to get all the information out there as much as you can about, you know, what is in this data set that you might be interested in. We require a little bit of work on the front end, and I'll explain a little bit more in detail in a moment why we require all this. But basically, you write up kind of a little metadata section that kind of gives an abstract, gives the methodology. It's almost like you're writing a little tiny paper, but what you're really doing here is giving all of the needed information out there so that someone can go and access this data set and know exactly what they're looking at and what they're using. There's also a data dictionary that it's very important to make sure that all of the variables that you see in the data set are very clearly defined so you know what you're actually seeing. And so this is what it looks like. One more sort of little pitch. This is like the carrot part of rather than the stick to try to get people interested in doing this. the data set itself is then citable. So we meant what's called a digital object identifier, a DOI, same as a paper would have, and that is persistent forever in perpetuity, and that makes your data set itself citable. It's on a CC BY license, which means if anybody does come and download your public data set and they use that, wonderful, they cite you, that drives your H-index. It's just like a journal article. So, okay, so that's basically how the ODC is set up. But why does this really matter, and what can we do with all of this shared data? Well, what we're really interested in is making sure that we're following what are called the FAIR data principles, these sort of guidelines that were developed outside of the realm of even neurotrauma. But basically what this says is if you're going to be sharing data, that data needs to be findable, accessible, interoperable, and importantly, reusable. And so the whole idea here is you have sort of legacy data sets, perhaps things that are sitting in file drawers now that can actually be useful to other people. And hopefully folks can generate new hypotheses. And in the end, right, the thing that we care the most about is generating, sort of new knowledge discovery. So I'll give a brief little kind of vignette here about what we've done toward this over the last few years. So starting again almost 10 years ago. Oh, first, we care a whole lot about this whole FAIR data thing. So we've written a whole bunch of papers if you want some light reading about how sort of the FAIR data principles and how that applies to both spinal cord injury and traumatic brain injury. You can enjoy that. So I'll explain just a little bit about one of the, I thought, kind of an interesting example from our work. So again, oh gosh, almost 10 years ago now, we had a postdoc working with us, Jessica Nielsen, who's actually here in Minneapolis now. And she, speaking of file drawer data, she went back and had access to the Mascus trial data. So those of you who are preclinical spinal cord injury researchers will remember Mascus trial was back in the 90s, basically a big multi-site trial to try to get some consensus and set up some sort of standards and norms around the way that we collect preclinical spinal cord injury data. And again, in that file drawer data, there were all these variables that were collected that were really never published. And so what Jessica was interested in was going and doing some really kind of sophisticated data-driven analyses that basically ask the question, how are all of these subjects related? What are the patterns of relatedness between the different subjects in this cohort? And also, what were some of the differences that were driving, what were some of the variables that were driving the differences that we were seeing in outcomes? And so, long story short, I won't get into the details of the math here, but what she was finding was there was a group of these animals that had the same injury parameters, but were recovering very differently. And so she was sort of baffled what could be driving that. And what she found was that it was actually mean arterial pressure that was being recorded at the time of their injury that seemed to be a major indicator, a major predictor of whether they, how these animals recovered. So that was a really interesting thing because it was not hypothesis driven, it was data driven, and it created a hypothesis that then we could take a little further. So fast forward a few years later, after we've Jessica and Adam Ferguson actually needed to get all the rest of this Mascus data, and so flew off and actually spent a while in a storage unit in Rutgers, New Jersey, digging through literal file drawers, finding paper records and things, because they're nuts. But what they did was basically they were able to replicate this finding. They went and wanted to see if this generalized to some of the other sites in the Mascus study. Found a similar issue here, where basically there was this sort of inverted U, where if their blood pressure was too low or too high, these animals were not recovering very well. There was sort of like this Goldilocks zone that seemed to be really a very strong predictor of how these animals recovered. Then within the last year, sort of building upon that, Abel Torres Espin, who still works with our group, but he actually has now set up his own data science lab at the University of Waterloo. He then went back and looked retrospectively. So we work right next door to the San Francisco General Hospital. I'm sorry, the Zuckerberg San Francisco General Hospital. Mark's always listening. I'm so sorry, bud. So he went back, looked retrospectively through patient records there, and combined that also with the Santa Clara Valley Medical Center. And then did some very fancy math, as he is wont to do. But suffice to say, I'll just quickly before I'll go back to that. You can see below here the published data sets from all of the retrospective records that he went back and looked at are also included. You can go on the ODC SCI and look at these data sets. But basically what he found, he did this really interesting topological sort of cluster analysis. This is groups of patients that he found, but basically it's a really interesting paper, but I just pulled out one particular figure that basically showed the same sort of findings that we were seeing before in the animal models, right? So we had a sort of early successful replication of what we were seeing that was sort of data-driven initially, and then eventually we take those hypotheses, we apply those to human data, and we see similar things. And this is wonderful, I think, for us because, again, we work side by side with the actual neurosurgeons, the real doctors, and we're on calls with them every week, and so we can show them data like this, and they say, oh, this is really cool. This might sort of change the way that they look at things and hopefully affect practice. So I'll just finish up with just kind of by the numbers here of our open data commons right now. I will say the spinal cord injury community has been pretty good about sort of coming together and getting behind this idea of data sharing. As you'll see in a second, the traumatic brain injury folks are like a little bit guarded about their data relative to the SCI folks. But right now we have 217 labs that are set up in there. You can see like almost 1,000 users. We are nearly, this was last, these numbers were last done, I think, in August. So we see that we have 45 public DOIs. Those are 45 public data sets that are out there and available. We are almost to 50 now. I think we have 49 and maybe a 50th one will come next week. So, we're getting, uptake from all around the world and all around the country. And as Matthew said, that, this is a really important, it really, to me, does not necessarily matter what the domain is. It could be spinal cord injury, it could be traumatic brain injury. What we're interested in is making sure we're collecting this data, making sure that it's really well annotated and we know what we've got, and then making sure the community also knows it's out there for them to use. This is the traumatic brain injury ODC that we also have, ODC TBI, looks very similar. So this is sort of where we're at so far. Right now, what we're working on for folks who care about stuff like this, as common data elements are being developed, sort of constantly worked on as those things develop, and there is some consensus around the community about what our common data elements are. We have the ability to map those to your data set, so you can upload your data set and then actually have little pull down menus that can then map your variable names to common data elements to make harmonization across data sets that much easier. We're developing some analytics dashboards so that you can come in, look at a data set, hit a button and get sort of some data visualization, just some kind of basic descriptives, as well as a little bit more kind of the data-driven multivariate stuff that we love to do. And then there's also this other initiative, Panorama, that we're working on, which is basically a new portal that will allow folks like us. We study both spinal cord injury and traumatic brain injury, and I think there are probably a lot of folks like that out there these days. And so we're creating a new situation where you can come sort of one-stop shop and be able to access both of these in one place. Finally, just one last thing. If you are a researcher out there who is applying for NIH grants and you have to write these data management and sharing plans, we have written up kind of some helpful templates for you. We also have office hours every Friday at 11 a.m. Pacific. I guess I missed it today because it's Friday, isn't it? But we have other people who are always there, where you can hop on Zoom and chat with us about, you know, what is all of this data sharing and how does it actually work. And I'll finish with that. Basically, I just want to say, you know, I think that sharing this data and making sure we have good data out there is going to really help with reproducibility, translation. Hopefully, you know, increasing that transparency is going to lead to new insights. And yeah, as long as we're handling our data, managing it well, and sharing things well, we're going to improve all those things like rigor, reproducibility, translation, stuff that we all actually care about. This is us. So we're, like I said, over at the Zuckerberg General Hospital, the San Francisco VA as well, which when it is not foggy is, I will argue, is the most beautiful view of any VA campus. We can chat about that, but and these are all the folks. We've got tons of collaborators and folks working all over the place in a lot of different areas for us. So I will end with that. Thank you.
Thank you, Russell. Really quick again, just as a context, I know Russell mentioned it. We also think this is really important, both the Open Data Commons, but also helping to facilitate it. It's time consuming. There's some effort involved. And if you're a PI, it's sometimes difficult to do that or find the staff to do that or the hours to do it. So one of the things we have done here in Minnesota is approach the state program and say, hey, can we look at mandating this for the state program, but also shake loose a few dollars to help, you know, add that in instead of asking the lab to just eat it, basically. And then the second thing, for those of you doing spinal cord simulation work, scientists, like I said, we're already working with these guys with one lab, which is the Darrow group. And that just came up because we've had a long-standing relationship and really just doing the preliminary sort of beta test to build out the architecture. What does this look like? But we're going to bring that back to the whole neuromodulation working group to say, okay, here's what we got started. Now it's time to weigh in from the rest of the field about how are we collecting the data? How are we profiling people? What are we measuring? What is important? That's the next step for this. So I just want to put that out there. Now, thank you, Megan. We program note, we flipped Megan and Leslie. There's another kind of electrical engineering work that doesn't involve spinal cord simulation, but uses electricity. And Sasha mentioned it earlier because Sasha actually has implants. Megan is here to talk about implanted FES strategies.
Thank you. Am I on here? Can you guys hear me? Okay. Yeah, thank you, Matthew. And I'll echo Russell's earlier comment. This really is a great meeting. I think this is maybe my fourth time speaking at it, and it's just a really fantastic collection of people. So I really appreciate having another opportunity to do this. So I represent the Institute for Functional Restoration. We have a mission to restore function to people with spinal cord injury by creating sustainable commercial pathways for neurotechnologies. One of the things that you're hearing about a lot today is just the challenge of commercialization. So my group is sort of focused on that pain point. So today I'm going to quickly talk about what neuroprosthetics are, how they work, and then give you some historical data. As we mentioned, this has been around for decades, and so we've learned a lot over those years. And then I want to talk a little bit about two clinical trials that are in progress that will allow people to have access to this as a research tool while we're developing the commercialization path. Okay, neuroprosthetics are also called functional electrical stimulation. And they take advantage of a well-known phenomenon, which is that the muscles below the level of injury in many cases can actually be activated with low levels of electrical current. So if you can activate and coordinate the activation of several muscles, you can create functional movement. And so the past several decades in a number of places, but in particular in Cleveland, we've looked at applying this idea to the restoration of upper extremity function, trunk stability, standing, stepping, bladder and bowel, respiratory, cough, et cetera. So we have a long history of this. I'll give a plug for my 2017 talk where I give a lot of in-depth data on some of those other applications. But today we're going to be talking about neuroprosthetics for upper extremity activation. Okay. So the first commercial product was the NeuroControl Freehand. This was on the market for a few years. It actually got through FDA approval. It had 8 implantable electrodes and an implanted receiver. And then the person would wear a transmission coil that would bring power and communication to the implant that rested outside the body. And then they would wear a joystick on their opposite shoulder. As they raise and lower their shoulder, they could open and close their hand. And so this product was actually FDA approved. You can see that the FDA approved indications was for C5 or C6 spinal cord injury, basically for people who are able to move their hand in 3-dimensional space and have intact lower motor neuron status. So the device was able to provide people with 2 different grasp patterns, what's called palmar grasp, which is used to hold fairly large objects, or lateral pinch, which is used to hold smaller objects. And the study that was used to support market approval enrolled 61 people at 15 different sites, including some sites in Europe. This is the demographics of the people who were enrolled in that study. It was predominantly C5 and C6 level injury. But as you can see, this is a device that is can be used by people who are many, many years post-injury. This does not have to be used in a acute setting. So let's see if I can get this to come up. Yeah, so basically our outcomes from that study, and this again was used to support the FDA approval, people were very, very successful with this. They were able to use the system in their day-to-day life, incorporate it into activities of daily living. We have a standardized pick and place test called the grasp release test that is used to evaluate palmar and lateral pinch. And, you know, people were basically everybody in the study was able to benefit from this. And this is really important. It's an important difference between where we were back then employing FES compared to where we are with epidural stimulation. We can test whether you're going to be a responder or not with this technology before you get the implant. And so one of the reasons why the success rate is so high is that we know before you go in that it's going to be successful. And so people had long-term usage out many, many years. I'll show you some examples in a moment of people who have decades of experience with their systems. So the NeuroControl system, the company went out of business. So, and by the way, they were the US distributor of the Vocare system that we talked about earlier. So the reason the Vocare is also not on the market is because when they went out of business, they took the Freehand and the Vocare system out of the commercial situation for the US market, which is unfortunate. So during that time that the Freehand was on the market, the Cleveland group went back to iterating on the design to try to bring an improvement on the Freehand system by making some of those external components more implantable. And so the system that they developed, which is the IST10 or IST12 system, improved in the sense that they had now 12 electrodes to work with. They had 2 recording electrodes that could allow you to measure and record voluntary muscle contractions and twitches that were used to control the opening and closing of the hand. This was intended to be Freehand too, except that the company went out of business. So this went back to our research labs and we were using it more for clinical research. So with the addition of these other channels, you can now provide people with new kinds of grasp patterns. These are just a few examples of things like, you know, you can position the hand gently to hold a sandwich instead of digging in with your fingers. You can use the pointer finger to scroll on screens. The cashew or popcorn grasp is a very light, gentle 3-point grasp that you can use to pick up small objects. So just adding those extra channels gave us a lot more flexibility in the kinds of things that we could do. So these are the kinds of study participants that were in our clinical trial. Just basically, we were able to report on 12 people, including 3 people that had bilateral systems. So we had 15 arms that we were studying. And these are some of their outcomes. So lateral pinch force, which was one of the measures that we take as a general rule, most people were able to double the amount of force that they could produce with stimulation compared to without. So this is probably the biggest functional improvement, is just having a stronger grip. And then looking at the capabilities of upper extremity function tests, where people are asked to do a whole bunch of different activities with their upper extremity. You can see that the green blocks represent places where their performance was improved with stimulation on. Yellow is about the same, and red would be a few cases where it was actually worse. But the point here is we had a lot of data. So one of the things you'll note is that neither of those two systems are commercially available. Freehand is off the market. The IT system was a research unit. We're no longer making that. So what are the options now for having access to upper extremity neuroprosthetics? So I want to talk a little bit about a couple of options that we currently have. So the Case Western Reserve University Network Neuroprosthesis is the 3rd generation system. This takes a lot more of the technology and puts it inside the body. So now you don't have to have an external coil for powering. The power supply is in the body. It's a rechargeable unit. And it's a modular system, which means you can sort of configure it to reach the different parts of the body that you're trying to activate with electrical stimulation. So right now we're studying it for both upper extremity and function and trunk stability. This is the demographics of the people that we have so far. Like everybody, we took a pretty big hit during the pandemic, and just enrollment kind of ground to a halt. But we have 7 people in the study right now. This is sort of the range of level of injury, time post-injury. And we have 5 people who have the upper extremity system and the trunk system simultaneously. And we have a couple people who only have the upper extremity system. So these are just some brief outcomes. We actually have a ton of data on folks, but we're still adjudicating some of it and analyzing it. But this is, again, the grasp release test is a standardized pick and place test that has 6 objects that people are trying to manipulate. You can see that before surgery or with stimulation off, on average, people were able to move 1 or 2 objects, but with the stimulation on, they could pick and place all 6 of the objects. It's important to note that an improvement of just 1 additional object is considered a clinically meaningful difference for this test, which is a validated test. So you all know Kim Anderson. She's an NNP user. She has a system for both trunk and for hand grasp, so she can turn the stim on her back to hold her steady while she's doing something heavy, like opening a refrigerator door, and then picking up a fairly complicated large object and hold it tightly and securely. So she's a daily user of the system. So in terms of like availability for this, we are enrolling in this clinical trial right now. We have 2 trial sites, the Cleveland Metro Health System, and also we are just standing up a new site in Philadelphia at Thomas Jefferson. If you want to search on that clinicaltrials.gov number, you can find our study, and that gives the whole list of all the inclusion criteria for the study. We're hoping to enroll up to 30 people, and we're currently funded to enroll 13. We're looking at hand grasp outcomes only right now, but people who get the system for hand grasp can become eligible for some of our other studies looking at trunk stability and other things. Okay, so now I want to sort of switch gears because, you know, one of the things that one of the painful lessons learned for our past history in Cleveland is just, you know, watching neurocontrol. You know, the researchers did everything right. They got it launched into a new company. The company started to sell for a few years, but then due to a whole host of different reasons, they went out of business. So it's really, really hard that ecosystem for commercialization is very fragile. And so for what we want to do next is try to really design something for commercialization to try to hit that target as our primary target. So to do this, we're trying to think a little bit more like Silicon Valley. You've all gotten apps that don't work so great, but after they're out there for a little while, people report on the problems and they get fixed. And so this idea of a minimum viable product is one way of doing design for a new product concept. The idea is that you push it out there and have users start using it and then have their experience shape what happens next. And by users here, we mean the people who are actually using it functionally for hand grasp, but also the physicians who are implanting it. This has to fit within their paradigm of care. Okay, so to get that underway, we did stakeholder interviews with just dozens and dozens of people. We talked to people with spinal cord injury, both people who had familiarity with FES systems and those who didn't. We talked to clinicians, surgeons, therapists. We talked to engineering people, business, industry, et cetera, to try to get a picture of what is this minimum product that we're going to try to actually commercialize. And so two really important requirements really bubbled up to the top. One is that this device needs to provide reliable grasp. It can't vary day to day. It can't be a source of frustration where some days it works, some days it doesn't work. And it has to enable people to do simple things like hold day-to-day objects, pencils, forks, that type of thing, a stylus, stuff like that, fairly securely so that they can actually incorporate that into their daily use. We also want it to be easy to implant. And the reason for that is kind of obvious, like, the patients themselves want an easy implant procedure, the docs want to have an easy implant procedure, but it actually sets you up to have a better experience with reimbursement if you can make sure that this device is going to easily go in. The hospital charges are going to be much less, and they're going to be more likely to want to buy it. And this is a painful lesson learned from our freehand experience, because with freehand, the surgery time was really, really long. Like they were doing concomitant reconstructive surgeries with the placement of the FES system. And so you couldn't even schedule another procedure in that OR that day. You were basically going to be using up the OR for the whole day. And this has been commented before by Nate. You can't have centers that are only doing 2 implants a year. That's going to kill you on the business model. You need to be able to get that implant procedure rate up higher on a per site basis. And so these are inflation adjusted calculations for what it would be costing hospitals to implant this today. But hospitals were taking a loss on the implant of the neurocontrol freehand system right from the very beginning. So that was one of the reasons why it didn't go so well. So with Key Group, what we're trying to do is, like I said, make it reliable, make it easy to implant. And so in our subsequent questions around design, we tried to figure out what grasp patterns we're going to be using, how many muscles, how many electrodes, how many incisions. All of this stuff is going to feed into the design. And so one of the biggest decisions was, this is only going to offer one grasp pattern. You're not going to get sandwich grasp and popcorn grasp. You're only going to get one. And everyone agreed, for the most part, everyone agreed that lateral pinch or key grip is the predominant grasp that people use in their everyday life for holding simple objects. The notable exception being our FES users who said, don't make me have fewer options, because they enjoy a lot of options for hand grasp, but they also understand that for people who don't have anything, having lateral pinch would be probably the most important option. Those are people with FES systems using lateral pinch in daily life. So our design decisions are that we are going to have 2 or 3 implanted electrodes, and like the freehand system, it's going to have an implanted receiver, but not implanted power supply. So there will still be external equipment worn outside the body, but that's our receiver design right there. It's very, very tiny. The whole thing could fit in someone's forearm. There would be 2 electrodes under there as well, and then there would be some sort of cuff-like device to communicate to it, to direct it with power and communication. And so how do we know that 2 channels is going to be okay? So we wanted to do a proof of principle study to see how this would stand up. And luckily, we have people that have 8, 10, or 12 channels of stimulation. So what we did was we brought some of them in and temporarily down programmed them to 2 or 3 channels and then measured their outcomes. So this is our proof of principle that we can provide useful function even with just a couple of electrodes. These are our study participants. you know, it's sort of heartbreaking to see, like, we really do have people who are 29-year users of the freehand system. This is a system that doesn't break. It just goes on and on. And we have with people with decades of use of the IST system as well. And so what we saw was that, once again, even with just 2 or 3 channels, we were able to generate about twice as much force as people would normally have without any stimulation. So this is going to become very useful for people in their day-to-day life. When we're doing the grasp release test, in this case, we only have 3 objects to work with because there's only 3 that are qualified for lateral pins. But you can see that people are improving in their ability to grasp and release objects. And if you could start these videos, please. You can start them all up at the same time. This is our freehand user with 29 years of experience with her freehand system, showing how this is one of the standardized tests. It's a simulated fork where you're trying to stab meat. And so it's like a plunger on a spring. And she's demonstrating that she can't actually do this task with no stimulation, but with 2 or 3 channels, she's able to generate enough force to actually pull it down and pass the test. And if you could start these 2 videos up, the second one. This is just another example of one of the standard tests. This just shows you how hard this test is. So again, this individual is a 26-year user of the freehand system with 3 electrodes activated. He can actually pass this part of the test that he otherwise wouldn't be able to do. And here's probably the thing we're proudest of. We had an independent group come and assess the reimbursability of this product and what it would cost for hospitals to purchase it. This is a big, big improvement in what was done with freehand. It makes us feel more confident that if we go down this path, we'll be able to actually deliver something that will be accepted in the commercial space. And so we are going to be starting a clinical trial. We haven't set it up yet, but we're hoping to start the clinicals in next year, probably summertime to fall of next year. We're going to be enrolling people with C5, C6 SCI. We're currently funded to enroll about 15 people. We're looking just at grasp outcomes, and the study will be used to support market approval for key grip. And when you come to Cleveland for research, we have fully accessible accommodations that can house up to 5 research participants and their caregivers. So we have a place for you to stay right next door to our research facility. And I want to acknowledge our funders. I think I forgot to put an INDS on here. They are funding the NNP study. And a special thank you to U2FP's advocacy to the state of Ohio for getting their SCI research fund up and going, because that is currently funding key grip. Thank you.
Thanks, Megan. So now we're going to hear from Leslie Morse from the University of Minnesota, Department of Rehabilitation Medicine. Oh, and by the way, I've stayed in that place. Yeah, the Z House, when our meeting was out in Cleveland. It's actually quite nice. Nice setup, very communal. I think I cooked dinner in that kitchen. So now Leslie's going to talk about clinical trial design, which think of this again, as we've been talking throughout the day about how research is done in the lab, how it comes out, how it connects on the other side. How are we threading these things together to make sense of them? And there's been a lot of discussion of late, I think in the last few years in particular, about how we do clinical trials. And so we thought it would be a nice way to kind of wrap up today, thinking about that particular question. So please welcome Leslie Morse, again, representing our title sponsor who wasn't here earlier that I mentioned. Now she's can welcome you all.
Okay. Thank you, Matthew. And yes, welcome, and thank you for staying to the very last talk just before the reception. So I really appreciate that. Go ahead and advance. Next slide. I have no disclosures or conflicts of interest that relate to the talk today, but I would like to acknowledge funding sources for other research that I'm not presenting Craig Nielsen Foundation, Neidler, and PBA. Next slide. Okay, so we know that the clinical trial is at the crossroads between scientific innovation and clinical implementation. And in fact, it is one of the gatekeepers, one of the, if you will, the barriers to moving things that are potentially beneficial and shown to be interesting and exciting in the lab into the clinical space. So we need these clinical trials. We need more of these clinical trials. We need them to be accelerated. We need them to happen faster in a more efficient way, and we need them to be successful, because without the clinical trial data, what we're moving into the clinic for obvious reasons may not have a very long lifespan. So it was a really nice paper, I think, in 2022 by Dietz et al. That looked at the clinical trial experience in spinal cord injury from 1996 to 2021. And there were some really interesting trends that came from this summary of this body of literature. Namely, if you look, the majority of these trials are very, very low number, with almost, the great bulk of these studies enrolling fewer than 100 and even fewer than 40 participants in their studies. The majority of the studies, let's say about 2/3 of the studies were completed, but less than half of the studies actually had results to report. And almost the majority, the entirety of this body of literature looked at one intervention per study. Next slide. So in spinal cord injury in general, we know that there are multiple challenges to meeting both enrollment goals as well as study goals. And so low injury incidence, injury heterogeneity, high cost of conducting some of these studies, prolonged study completion time, lack of consensus defining what is a successful treatment and what is not a successful treatment, and resistance to control randomization. And I am also guilty of this. It's really difficult to design studies where you know that half of the group is going to be receiving no intervention or no effective intervention. So outside of spinal cord injury, there are limitations. Additionally, please, next slide, to the randomized control trial, which is considered, the conventional randomized control trial is considered the gold standard in clinical trial design, but there are limitations there as well. High cost, again, time to completion, and a very, very high completion rate, high failure rate. Importantly, the conventional randomized control trial has a very inflexible study design. And by that, I mean that you have all of the parameters established at the beginning of the trial, and then you move forward according to those predefined parameters, irrespective of new information that may come to light either in the greater scientific community or as a result of your findings. Next slide. MJ et al. Had a nice summary in 2020, again, of clinical trials, and specifically adaptive clinical trial design and how it's been applied to spinal cord injury research, or actually not applied to spinal cord injury research up until this time. Narrow in scope, few in number, immediate application of several adaptive methods would offer an opportunity to greatly accelerate the rate of translation of important findings to clinical care. Next slide. So this is really, really, really telling and disappointing. Considering interventional trials across all disorders, the average total time to progress from a conventional phase 1 study to regulatory approval and market introduction is 12 years with a success rate of 9%. So the field waits 12 years for a success rate of 9%. Next slide. So this talk is really a call to collectively bring our expertise, our interests, and our skills together to apply what is already being applied in multiple other scientific domains to spinal cord injury research. I think it's really critical, important, and the time is now that we think about collaborative research that takes advantage of adaptive trial design, but specifically platform clinical trial is something that's underutilized in our field and may have potential to really accelerate advances. So what is the platform clinical trial? It is a randomized clinical trial that utilizes simultaneous comparisons of multiple intervention groups against a single control group. There is a pre-specified interim analysis plan. So there are at least one time point or perhaps multiple time points throughout the conduct of this trial where you stop, look at the data, and then make key decisions about how the trial may progress going forward. And that could be the introduction of new interventions. Perhaps new potential therapies have come to light and would benefit from being rolled into this platform design. So new interventions that come to being after the trial has started can be added using a master or core protocol. The control group and the standard care provided can also be updated during the platform trial as interventions are shown either to be effective or ineffective over time. So the goal of a platform clinical trial is really focusing on identifying treatments, not evaluating specific interventions. And so taking a pool of information and quickly identifying what may be a viable therapeutic intervention that can be moved forward. Next slide. Okay, so there are multiple advantages of the platform trial design. Multiple treatment candidates. It's most beneficial when there are multiple treatment candidates available. And so that is a potential limitation and where our field can become more organized and more specific or targeted about the type of research that we want to advance and when we want to advance it and how we want to advance it, meaning that we can collectively increase the bandwidth in specific targeted areas that may benefit from application of a platform trial design. Platform trial allows you to quickly add or discontinue an intervention based on new evidence. And one of the key elements of this is the efficient use of trial infrastructure. And I'll talk about that in a couple of slides. Also very, very important and very applicable to spinal cord injury research. It allows the reduction in the number of participants required because, again, you're using shared control groups across multiple interventions. So obviously, this can accelerate discovery. It can use networks and multiple trial centers to conduct studies to reduce costs and increase the number of interventions that can be tested with the same or a lower cost compared to an individual conventional randomized control trial. Next slide. So a couple of key differences between the conventional randomized control trial and the platform design. In a conventional randomized control trial, the data are analyzed after the last participant completes the follow-up. There is a fixed sample size and a defined endpoint, and there are typically two groups, an intervention versus a comparator or a placebo group. There can be multi-group or factorial randomized control trials that also assess multiple interventions, but the interventions are defined at the start of the trial, and that typically doesn't change. As compared to a platform trial where the platform trial is designed to continue for a long period of time. So the overall structure of the study continues and has a rolling kind of cadence to it, but it allows the introduction or the backing out of interventions at multiple time points throughout the study. There are pre-specified interim analyses that allow for adaptations. So you can have stopping criteria for either futility or superiority of an intervention. Next slide. So this is also an adaptive trial design in that the key trial characteristics evolve over the course of the study in response to new information or evidence that's being accumulated. And this can impact decisions around randomization proportions, sample size, and treatment groups. Again, there are pre-specified rules governing these changes, how the changes are made, when the changes are made, and why the changes are made. And they're based on the scheduled interim analyses. Next slide. And this is a schematic of what a potential platform trial might look like, where you have concurrent and non-concurrent treatment groups and potentially a control group. So you see in the first wave of the trial, there's a common control or a placebo being tested against two treatment arms. And then in the second wave, treatment 3, 4, and 5 are introduced using the non-concurrent placebo as the comparator. Okay, so I just use concurrent and non-concurrent controls. That's a key element of the platform trial. Obviously, if you're adding interventions, it's not necessary to add a control group every time you add an intervention. So the concurrent control is randomized and studied at the time of a specific intervention group, and the non-concurrent is a group of control participants who are enrolled and studied prior to a specific intervention. So in this case, treatment 6 and 7 would be non-concurrent to the placebo. This has some considerations for prognostic balance, which is the goal of randomization, so it can introduce some bias into the study design. But this can be addressed. Also, I should add that this is more of a concern in rapidly evolving diseases or states such as COVID or influenza trials, where a non-concurrent placebo may have less import 1 year later when many things about the course of the disease have changed. But nevertheless, you can adjust or address prognostic balance in the statistical analysis, considering temporal variability and other confounders that might have an impact, thereby minimizing prognostic imbalance. When we report our findings from a platform clinical trial, the trial should report if a concurrent or a non-concurrent control group was used in the analysis. And obviously, for a long duration study, such as a platform trial, where you're basically keeping this trial open for a long period of time, in some cases years, multiple publications will come out of a study such as this. Next slide. Another important point, the concurrent and non-concurrent controls are very different from historical controls. And so oftentimes there's an impulse, particularly in this field, to say, well, you know, we have multiple barriers to enrollment, and we didn't meet our goals, and so we're going to use this historical control group from a large data set of data that were collected, you know, in another setting. Historical controls don't have the same scientific robustness as concurrent and non-concurrent controls within a platform control study, because the platform trial allows for all participants in the control group to have been enrolled under the same inclusion and exclusion criteria and for standardized data collection. So really, this is higher scientific quality of data than a historical control. So it is possible that you may have, over the course of a platform trial, an evolution of the trial itself or the interventions that are being tested, and the eligibility may vary. And in that case, multiple control groups would be required. If participants are only eligible for a subset of interventions, the control group for that analysis can only include participants eligible for the intervention. And so in that case, you would need to fold in additional placebo groups to be able to address the appropriate comparator. Again, I've mentioned interim analyses are really key in the decision making over the course of a platform trial. It is an analysis of clinical efficacy conducted prior to the last participant completing the trial. It can lead to early stopping for futility or for statistical superiority, where the benefit exceeds a predetermined threshold. When an effective intervention is identified, the control and the standard of care can be updated in the platform. So let's take neuropathic pain as an example, or a pain study. If, let's say, an anticonvulsant is identified as superior to other arms in the study, that potentially could become the standard against which the other arms are tested going forward in the platform. Futility, when the intervention demonstrates little or no chance of benefit, even if multiple additional participants were added to the study. This trial design allows you to just take that arm out and focus resources on other interventions that may have benefit. So I said that we can stop for superiority. So you set predefined thresholds for what would be a superior response. But again, these are small studies, small sample size, and that can increase error. And so the thresholds for superiority are typically much more conservative than in the final analysis. And, so another element of adaptive trial design in platform trials is response adaptive randomization. So allocation ratios can be adapted based on the interim results. So you can actually randomize more participants to treatment with a favorable interim result so that you have more participants running through that arm and the opportunity to definitively demonstrate that that intervention is effective. The allocation ratio for the controls would remain fixed in that case. Okay, one really critical component of the platform trial is clinical trial simulations. So these simulations are basically modeling of a virtual patient population using large data sets that exist and introducing into the statistical analysis. Yes. Oh, I'm sorry. I'm so sorry. So okay, here we are. So one more. So clinical trial simulations, you essentially take a virtual patient population from an existing large data set, and you run modeling to try to predict how participants may respond to various interventions based on physiologic characteristics or other characteristics, demographic characteristics of the virtual population. This can be repeated. So typically you would run a simulation at the beginning of a platform trial to set predefined standards for how the trial would run, and then that can be repeated after multiple interim analyses are conducted at multiple time points. I got a 5-minute warning. I've got about a minute, and so I'm gonna, if you could advance to slide 25 for me. Back one slide. So this really visually is the benefit of the platform clinical trial. And it is in the savings of cost, infrastructure, and time, right? So for each one of these interventions to run a conventional clinical trial, you have all of the elements that have to be set up individually and then repeated each time you move on to another intervention. And so next slide, platform allows you to basically create and sustain the infrastructure for each of these individual randomized controlled trials, put them into the same space and accelerate essentially findings from each of these studies. And with that, I think I'll stop.
Thank you, Leslie. Yeah, Russell, come on up. Megan. Oh, you need chairs. All right, we're almost there. Questions before drinks?
Get your drink tickets out now.
Susie, that was quick. Yeah, here.
Oh, thanks. Okay. Okay, come over here a little more. So I come with me. Okay, so I have two questions, if I can take the liberty. So the first is about, the data sharing. I mean, it's really powerful and important. It's difficult logistically, I think, for researchers. I was thinking to myself, I probably have data in drawers that could fill this room. And I'm embarrassed to say that, but it's true. And I was listening to you and I'm thinking, okay, how do we do this? Because you may have a little bit of funding, but it may not be continuous. What you really need is a full-time team person, at least, who knows how to do this and is just constantly doing it, right? So, but I heard you say, I think I heard you say that people went somewhere and got someone's data. So I was thinking this would be fabulous if that infrastructure could be built from the knowledgeable people who could then come in and interact with the labs. Did I hear that correctly? Or
Yeah, that was one particular instance very early on, before as we were setting up, even before the site, that was sort of like the early episode of data archaeology that folks are doing. I mean, I think it's a wonderful idea to have sort of, and we actually have a data librarian now who works on our team to sort of help with all of this. But we currently aren't going out and doing that, digging through people's file drawers.
you can come to mine.
But we could. I mean, yeah. I mean, that's.
The original one was it kind of made the case.
Exactly,
Made
and
the case for why
Yeah.
this is.
that's right. That was sort of that it's kind of established, and it was also a way, and Adam's given talks on this too. It was also a way to sort of show like we should not be having to do this all the time. Hopefully, going
Yeah.
forward in the future, let's not be doing this. But to your point, I think it's a great idea.
Because you would have the experts, because I'll tell you right now, just with what our requirements are, it's intimidating for the team. They don't know how to do it. They don't really understand it. They upload things, and I think if there was more of both a collaborative and then there's the experts, right? You just come in and be so much more efficient for researchers. Of course, you'd have to invest too, but maybe that
Certainly,
would reduce
yeah,
the financial and time risk.
definitely. And I think that will come to that. This is definitely thank you for that. This is one of our charges too, I think, is getting the word out to the community. As we build this up, what I'm hoping for is that people will start to see the benefits and can see a sort of a return on their investment of like, boy, why do I have to do all the, even outside of the file drawer issue, it does take a little activation energy in order to get people to do this. They don't want, for the most part, they don't know how. I mean, and we work with them. That is one thing. Like, we're pretty much at right now sort of a concierge. Hey, if you've got issues, if you were trying to publish a data set, you hit that button to say, I would like to publish this data set. Our team goes into action and we work back and forth with you, sort of the way if you're publishing a paper, there's sort of an editorial board. But I agree completely that that would be a great thing to have out there, to be able to salvage some of this legacy data that's probably really valuable.
Rob?
Hello? Okay. The theme that I think about kind of a lot when it comes to research, and especially today, it started this morning when the funders were up, and working for you, Dr. Morse, in research, and I see the cogs in the wheel, and it's always like been disgusting for me, but it is necessary. But like as somebody living with a spinal cord injury and a high level and no independence, my currency is time, and I'm losing time. I'm losing, from things I want to be doing. And, I keep emphasizing we need to do this faster and, more efficient. And, but, this kind of alternative clinical trial design, I think, allows I think it's something that funders should be more open to, and investigators, and you know, being able to modify interventions as you go when you see something that works or doesn't work. But also, you know, this time lag of, okay, you have regulatory, now you have to do the study, which is maybe a third of the overall time from a grant cycle. And then you publish, which is another 6 months to a year. And then you can write a grant to get the next level, which is another year. I mean, so if you can do these things, do interim analysis and apply for funding as you're going and modify, I don't know. I think it's a great idea.
So Matthew, if I can, thank you, Rob. So one thing I didn't mention during my talk is that one shift in the way that we are currently funding clinical trials would be to invest more in the discovery preliminary pilot phase, and then set up these platform trial networks where things can be rapidly run through an existing infrastructure. And so one of the limitations, perhaps one of the reasons that we don't do platform clinical trials that frequently, is that we don't have enough viable interventions to be testing simultaneously. And so we need to increase the number of potential interventions in the discovery space so that we can move them through these efficient trial designs.
Anita.
Hi, and sorry, I forgot to introduce myself earlier. So I am a middle-aged woman with brown hair, over 25 years of experience living with a spinal cord injury up in Toronto, and also doing my doctorate at U of T. So my question is for Russell. I understand the benefits of what you're doing for data that already exists, that hasn't been open to the public. But going forward, would it not just simply be easier to have journals mandating that authors openly publish their data? I mean, you know, you can add it in as a supplement to your articles. I mean, it would be a much easier process, I think, than this. So I'm just curious, what are your thoughts? Like, what's the benefit? Am I missing something?
Yeah, so two things. I agree completely that would be wonderful if the journal, and a lot of the journals are sort of now taking up that mantle to say, of course they've had for years, you know, the little statement, you need to make a statement saying, oh, I'll make the data available if somebody asks for it, which is sort of like, you know, not really very rigorous. And so I love the idea of journals starting to be more direct about, you need to have, there are some journals now that say, you know, you need to have the data set needs to be accompanying the paper. The one thing I'll say about what we provide apart from the journals is, and this isn't all journals, but sometimes, so for instance, if you have your data set as a supplemental file on a journal website, it is not quite, it's susceptible to link rot. If you're familiar with this term, it isn't always necessarily going to be there. The difference is, and I'm not saying we are the only game in town as far as minting a DOI for a data set. So if a journal says, oh, we want to attach the data set, and they're willing to mint a DOI for it, I think that would be wonderful. The thing that the DOI does for it is it creates a persistent link that outlasts the journal. It outlasts us. If we somehow ODC SCI goes away tomorrow, that DOI will persist. It's backed by the University of California libraries. So as long as the concept of the library exists, then it's there. So it kind of takes some of that burden off. But in general, I totally agree. You know, we're interested in making sure that the data are out there and fair. You don't need to come to us. But I will say that sort of extra bit that we do, that it can be a little onerous for other people, I think in the end makes the data that much better. There are plenty of repositories out there that don't require much of anything in the way of metadata. And I would politely say that those data are difficult to find and they're not accessible and they're definitely not interoperable. And in the end, they're completely unable to be reused. So there's a reason, you know, that why we're kind of putting our foot down and like, we want to be the ones that are adhering to these FAIR standards. But sure, I think if a journal wanted to also kind of take that up, I think that would be great too.
Jesse.
Hello there. My name is Jesse. I got 3 years injury, and to kind of echo Rob's point, we are running out of time like everybody else is. So I did a clinical trial for a year, the randomized trials, and I totally thought they were clunky and slow, full of holes. Somebody can't make it in that day. It just got pushed ahead. The platform style is sounds great. Is there a hybrid there that might be possible with platform and with our AI, with artificial intelligence? I'm not a big fan of all of this kind of crazy new technology going on, but obviously it is really helping us. And so if we do have all this metadata out there and we got AI, can we try to really speed this along?
I know that your comment was probably more up to the clinical trial design, but I will say that your frustration around the slowness is felt by a lot of people in research too. And coming from a group that for decades has been working in an area and hoping to commercialize something and get it out there and available, it takes making that concerted effort to do it towards commercialization. I would be interested on the novel trial design to come back to some of the things we've been talking about today. Epidural stimulation is a great example of something where you turn the device on, you turn the device off. You don't have to have a control group per se. Each person can act as their own control. And I would be curious to see how sample size drops down if you have a model that's based more on that. So you don't have to randomize people to no product.
One thing I will say is it would be great if we didn't have, obviously we're in a system and an environment of limited resources, and leveraging our resources towards forced collaboration instead of competition. So 2 groups doing epidural stimulation trial, limited funding at NIH, really investing in forced collaboration between multiple groups in the field, right?
Following a standard protocol, like following the same protocol, having the same outcome measures, looking at having the same inclusion criteria, like, maybe publishing, I've seen this done lately, publishing the clinical trial design so that people wanted to follow your trial design, they actually could. That would get people off to a start of at least being able to do a comparable study so that later you can pull the data.
Yeah, over here, Murray Blackmore. Can people hear? Oh, can you hear now? Oh, much better. This is still on the platform design. Hopefully this will be quick. I get the concept and I think it's great, but when I look at the landscape, I see companies with their individual lead compounds, Exonus and NerveGen and so on down the line, and they're not going to pool their resources for a platform trial. So can you clarify what is the entity that would be running this? Who's got IP on 6 different shots?
So ALS is the leader, in my opinion, in platform clinical trial design in at least rehabilitation, or at least it's the literature that I'm most familiar with. And so it's really a very nice, in the ALS space, it's a very nice collaboration between academia. So it's based at MGH, at Harvard, and in industry. And so I actually, the first time I had heard of the platform trial design, one of the lead investigators gave a talk and basically described it as the trial is a house, and there are always rooms in the house that are open, and people come to us with their ideas and we slot them into our house. And so there are ways to incentivize industry and academia for the goal of accelerating research and progress and finding a cure for, you know, a horrible disease like ALS that is, you know, rapidly progressing. And so I think there's a bit of motivation to rise above and do good for the field, but also there has to be concrete benefits and incentives, and they exist. We can figure out how to work that out. Certainly somebody has to own the house and run this, and you know, and that's the infrastructure. So somebody with expertise, interest, resources, and the ability to bridge multiple stakeholders is the person who should probably be raising the platform trial, you know, probably in collaboration with industry leadership as well. That's how I see it happening.
Last question.
I'll be quick. Sam Efford, I'm a software developer in the data science team at Ancestry. And there's so much of what you guys are talking about that overlaps with the kind of work that I do. So just I don't know if it's questions, but comments. I don't know if we've explored crowdsourcing, either crowdsourcing the data collection and talking with Marco from the Reeves Foundation. Apparently Parkinson's communities are ahead of people wearing watches and smartwatches and crowdsourcing that data collection. And then the other thing is maybe some gamification of the data analysis, where these devices and the research studies were spending most of their time trying to tweak the software, and artificial intelligence can do that better than a human can do. But you got to hire the data scientists. Maybe there's not funding, but you can crowdsource it. A few little $10,000 awards on Kaggle, which is a contest website for data scientists, potentially could provide the software or the artificial intelligence models to move some of this stuff forward. So anyway, other conversations, hopefully. Yeah.
Okay. Thank you. Yeah, thank you, Susie. It's been a long day. I appreciate your patience, especially going a little over time with our technical stuff and my terrible moderation. But so thanks for hanging in there. Tomorrow we're gonna pivot a little bit, move in a different direction, but continue, I think, a very similar theme throughout the conversation. But right now, you have some tickets in here to go get a drink, if you like, of any kind. And you can go right through here, and there's some hors d'oeuvres waiting for you. And you can continue the conversation with everybody. Yeah.